Konnichiwa guys, welcome to our channel Loops of Biology. It's been a long days we are learning biomolecules, but still there is new interesting topic about it. That is nothing but your nucleic acids. So if we are talking about what are nucleic acids, so nucleic acids are comprised of your DNA and RNA. So both of this nucleic acid are very important whenever we are considering that inside a cell. So if we are talking about DNA, this DNA is present within your nucleus, while if I'm considering about RNA, that is present inside your nucleus as well as cytoplasm. So nucleus plus cytoplasm. And every of this molecule has its own function and own properties. So if we are talking about common factors of this nucleic acid, so this is basically, if we are considering monomers, so monomers are basically of nucleotides. So if we are talking about nucleic acid, the monomers are nothing but your nucleotides. So these nucleotides are further divided. These nucleotides are further divided into your ribose sugar. Then there is going to be a presence of nitrogenous base, and there is going to be the presence of phosphate group. So these are basically the three components which are present inside your nucleotide. So what if we are talking about ribose sugar? This ribose sugar is nothing but your aldoses. Are you remembering my prior videos? So aldoses are nothing but a sugar that is containing aldehyde as main functional group, and this aldoses is of 5 carbon. So this ribose sugar is of 5 carbon. While if we are considering nitrogenous bases, this nitrogenous bases are basically comprised of 5 different nitrogenous base that contains heterocyclic nitrogen containing ring. So what do you mean by heterocyclic? If we are talking about a normal cyclic structure, all right, if we are talking about a normal cyclic structure, that will seem like this. Whenever the every bond formed within the structure is between your carbon. But if we are breaking this particular chain by adding one extra element inside it, which is inorganic element, then that particular ring will be called as heterocyclic ring. So here the particular inorganic element used is nothing but your nitrogen. So those are nothing but your nitrogenous bases, and we are well known about the phosphate group, that is nothing but phosphorus attached to your one oxygen with double bond and 3 hydroxyl group through single bond. So if we are talking about the phosphate group as such in the form of ion, that will be looking as PO4 minus minus minus, that is PO4 3 minus. So in next part, if we are joining all this monomer together, then we are going to form your polymer. So in this case, the polymer is nothing but the joined parts of monomers. So there are all monomers are basically joined together through one bond to form a polymer. Like we saw that in case of carbohydrate as glycosidic linkage, in case of lipid that was your ester linkage, while in case of protein that was peptide bond. So similarly in this particular case, the polymer of this nucleic acid is known as polynucleotide. It's as simple as that. If we are going through a name, so poly means multiple, while nucleotides are nothing but your monomeric units of this particular nucleic acid, and these are joined together. They are joined together by phosphodiester bond. So they are joined together by phosphodiester. This is a special type of bond which is present commonly inside both the nucleic acid, that is DNA and RNA. Coming to the next part, that is what is nucleotide. So as we have seen, the nucleotide is comprised of three different parts. So first one was nothing but here ribose sugar. Alright, the ribose sugar was belonging to your case of aldoses. So what was aldoses? That is nothing but aldehyde as main functional group. As main functional group. And this belongs to your pentoses as well because of the presence of 5 carbon. So it is belonging to a category aldoses, while based on the carbon number it is belonging to your pentoses category. Next case is nothing but your nitrogenous base. So there is going to be the nitrogenous base, and third case was nothing but your phosphate group. So there is going to be the presence of phosphate group. However, if we are talking about nitrogenous base, there are two different categories. So the nitrogenous base based on all the nucleotides, right? Based on all the nucleotides belonging to your DNA as well as RNA. We are not specifically talking about any of the separate nucleic acid. So whenever I am considering nitrogenous bases, there are two categories as purines and pyrimidids. So if we are talking about purine, as we have seen, the nitrogenous bases are made up of heterocyclic ring that contains nitrogen inside it. That's why they are called as nitrogenous bases. So if we are talking about purines, there are two heterocyclic rings, and that if we are considering about pyrimidine, there is going to be only one heterocyclic ring. So there is going to be presence of only one heterocyclic rings. So what are the nitrogenous bases now? So if we are talking about purines, so these purines are nothing but your case of adenine and guanine. And that if we are considering pyrimidines, those are containing your thymine, cytosine, and there is going to be uracil. So this uracil is specifically used inside your RNA, while if you are talking about thymine, this is specifically used inside your DNA. The cytosine is represented by C, guanine represented by G, and adenine represented by A. So if the particular structure has only two parts, that is ribose sugar and nitrogenous base. So if there is only 1+2 condition, that is nothing but your ribose sugar and nitrogenous base, then this structure is called as nucleoside. And whenever I am adding phosphate group, so whenever I am adding phosphate group inside nucleoside, then it becomes nucleotide, and this NT is official short form for this nucleotide. So in the upcoming videos, if I'm anywhere using NT, this is nothing but your nucleotide. So what are the types of nitrogenous bases and sugars? So if we are talking about basic as a nitrogenous bases, so there are two categories as we have seen, that is nothing but your purines and pyrimidines. So purines were loaded with your two heterocyclic rings and belonging to a category of adenine and guanine, and that if you are considering pyrimidine, that contains only one heterocyclic ring and that contains thymine, cytosine and uracil. So if we are talking about the bonding pattern of this nitrogenous bases, the bonding pattern, then there is always a binding between purine and pyrimidine. Purine and pyrimidine bond formation. Bond formation. Now, if we are talking about the bonding pattern, then adenine can bind to thymine through double bond, or adenine can bind to uracil through double bond. So basically, inside case of RNA, the thymine is replaced by uracil. And that if we are considering cytosine and guanine, then guanine will be binding with cytosine through triple bond. And these bonds which are formed between the nitrogenous bases, the bonds which are formed between the nitrogenous bases are purely hydrogen bonds. They are not covalent, they are not ionic, there is no coordinate, but only hydrogen bonds are present. And the hydrogen bonds are going to form whenever the hydrogen is coming in close proximity to the electronegative element. Alright, so if we are talking about what are the types of sugar, so there are two types of nucleic acid, that is DNA and RNA. So if we are talking about ribose sugar as common, so sugar is same, that is ribose sugar, but this ribose is present inside your RNA as a native structure. So this ribose sugar as it is without any changes, it is present inside your RNA. But if you are talking about DNA, then DNA contains deoxyribose sugar. So what is deoxyribose? So as per the name suggests, deoxy means removal of one oxygen. So deoxy is nothing but removal of one oxygen from a ribose sugar. So this ribose sugar will look like this. So if we are talking about this particular structure, so ribose sugar seems to be like this. So there is OH, H, OH, H, there is again OH, H and CH2, OH. So this is your general ribose sugar structure. So as the name suggests, that is deoxyribose. So we have to remove out one oxygen. We have to remove out one oxygen from the second position of the sugar. So there is going to be just a presence of hydrogen. So as we start to number this particular structure, there is 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. So there are total 5 carbons in which there is absence of one oxygen atom on the second prime carbon of the sugar. So there is going to be absence of oxygen on 2 prime carbon. So this dash on the number represents prime. Please do not pronounce it as dash. It is prime. So that is 1 prime, 2 prime, 3 prime, 4 prime and 5 prime. That if we are considering in case of ribose sugar, then there will be the normal structure without removal of any oxygen molecule. So there is OHH. On second prime there will be OHH, third prime OHH, and finally there is going to be your CH2OH. So this will be your structure for considering a ribose sugar. So moving on to the next part of this particular nucleic acid, then why deoxyribose sugar is present and what is basically the ribose sugar doing inside a particular structure. Whenever I am considering ribose or deoxyribose sugar to be a part of DNA. This sugar is basically helping for attachment of phosphate group attachment of phosphate group and nitrogenous base. So whenever the sugar is going to be formed, this sugar helps for attachment of phosphate group and nitrogenous base. So where this particular bases and groups is going to be attached. So on 5' end of the sugar is basically the attachment of phosphate group. While if you are considering your nitrogenous base, so that is going to be attached on 1' end. So 1' end of the sugar is basically helping from attachment of nitrogenous base. However, the 3' end of sugar that contains your OH group, that is hydroxyl group, this is attached to next sugar. This is attached to next sugar molecule, that is nothing but your nucleotide. So that is attached to the next nucleotide. And this ribose sugar is very important as it forms a backbone of structure. It forms a backbone of structure. So it is keeping the particular structure stable by forming a bond and a chain of nucleotides. Is that clear? So next part is your phosphodiester bond. It's slightly tricky, guys, but I hope you will understand this particular part. So if we are talking about your structure of nucleotide, so there is going to be on 1' there is either nitrogenous base. What are nitrogenous base you have to add? On 5' there is going to be the presence of phosphate group, and on 3' there is oxygen with hydrogen molecule. Same structure is present on the bottom side of the sugar. So if I am talking about next nucleotide, there is again nitrogenous base. There is again OH group on 3'. Here if I am considering the deoxyribose sugar structure, then there will be H structure, and there is going to be the presence of phosphate group. So I am representing this phosphate group as OH and OH. So if we are talking about this particular structure, then again I am able to see two hydroxyl group in the close proximity. So from which I will be able to remove out your water molecule. So there will be removal of water. So this water will be removed out, and new structure, and the new structure is basically looking like this. So whenever I am considering a new structure, there will be a presence of sugar and oxygen attached to phosphate. This phosphate is going to have this kind of structure, oxygen, and there is carbon. This carbon is further attached like this, and finally the structure will be done. There will be nitrogenous bases on 1' of the region. So now the new structure which is formed between the two nucleotide, if this is your nucleotide 1 and this one is nucleotide 2, then the bond which is formed, between your phosphorus containing hydroxyl group and The 3' hydroxyl group present on this particular structure of nucleotide is nothing but your phosphodiester bond. Is nothing but your phosphodiester bond. So this bond is basically called as your phosphodiester bond. Got it? So if we are talking about this structure, let's first somewhere talk about how phosphate is attached to the carbon. So if there is CH2OH, this is your 5 prime. This is your 5 prime carbon atom. Then there is again OH, P, double bond O. There will be OH and OH. So in this case here, this particular structure is removed out. So in this case, you won't have this hydroxyl group. Got it, guys? So this is your phosphodiester bond. I hope this is clear. Why it is called as phosphodiester? Can anyone guess? As we know, what is ester linkage? That is C double bond O O R. Right? So in this particular case, there is a presence of phosphate group. So this structure, if we opened it up, it looks like OC. There is a presence of phosphate group with double bond O, like there is a double bond on carbon, then oxygen, and then again carbon. So there are two ester-like linkages mediated through one element, that is nothing but your phosphorus. That's why it is called as your phosphodiester bond. Got it, guys? So this was your phosphodiester bond. Coming up to the next part, what is disadvantages? What is the disadvantage of ribose sugar over deoxyribose sugar? As we have seen that deoxyribose sugar was your part of DNA, while your ribose sugar was a part of RNA. Now, the DNA is a genetic material. We have been learning through your 5th standard that DNA is acting as your genetic material, and genetic material has to be stable. So basically, DNA has more stability. DNA has more stability than RNA. So DNA is more stable than your RNA. So why is that? That is completely because of your deoxyribose sugar and the structure of DNA, of course. So whenever I am talking about deoxyribose sugar, the structure represents that there is absence of 2' hydroxyl group. 2' hydroxyl group in the ribose sugar. So whenever there is 2' hydroxyl group is absent, it is impossible to form intramolecular interactions. So there is no intramolecular binding. There is no intramolecular binding, and specifically 3' hydroxyl group is in the formation of which bond, guys? That is nothing but phosphodiester bond. So it was completely involved inside the formation of phosphodiester bond. But if we are talking about ribose case, so whenever I am talking about your ribose sugar, so ribose sugar has presence of 2 hydroxyl group in close proximity again. And whenever there is a presence of hydroxyl groups in close proximity, what we are going to do, that is nothing but removal of water. So after the water is removed, this kind of structure can be able to form. So there is a disadvantages of ribose sugar over deoxyribose sugar, that there can be intramolecular binding. I hope you are well versed with intra and inter word now. So there will be intramolecular bond is going to be formed, and this bond stops or halts your ribose sugar to form the next set of bond as through your phosphodiester bond, and this will stop or destabilizes the structure. So this destabilizes structure, and this is seen in case of RNA. That's why your DNA is more stable than RNA, and this is the only disadvantage of ribose sugar over deoxyribose sugar. I hope you clear with this particular concept and liked this particular video and understood what are nucleic acid actually. So this was your basics about nucleic acid, and in the next video we will talk about DNA and RNA, guys. For now, please do not forget to like, share and subscribe to our channel. See you in the next video. Bye bye, guys.